All right. Hi, everybody, and welcome to the webinar. I'm Ian from AISquared, and I'm joined by Mahmood from John Snow Labs. And in today's session, we're going to walk through kind of two thorny challenges that are facing clinical decision makers. So the first is, how do we turn an ocean of data into kind of clean, synthesized insights that can be actually useful to the decision-making process rather than just adding noise? And the second is, how do we take those insights and deliver them kind of where, when, and how they need to be in order to actually augment clinical workflows? So with that, I'll turn it over to Mahmood.
Thank you everyone for being here. Over the next few minutes, I want to show you how to automate systemic literature review with medical grade AI and how you get from an evidence search to a structurally cited drug insight so you can later analyze that and get some insight from that data. The tool is called Literature Review Agent, and let's start with the problem first of all. So the problem is evidence synthesis is essential and it doesn't scale. And that's the whole issue. And it's larger than it first looks. So the foundation of evidence-based medicine is used for clinical guidelines, for example, or to set synthesizes full body of trial evidence and instead of single studies. Also for drug evaluation, there's some similar solution there. For efficiency, comparator endpoints, and adverse events emerge only across many trials, and regulatory and HTA decisions and submissions. And also reproducibility is another key here. So you have to ensure that it's transparent and criteria driven so someone else can follow it and reach to same conclusion as you and verify the findings. So, which is why a complete, unbiased, and reproducible read of literature is non-negotiable, and decisions are as good as evidence behind them. Uh, the main challenge here is the challenge of scale, and the evidence outpaces people who must read it. For example, on PubMed, which holds more than 37 million records, over a million new paper lands every year. And to answer one question properly, a team reads hundreds of thousands of abstract per review, and that takes weeks to months to complete. So by that time the manual review is published, the field has often moved on, and the review and the result might change and expire. So the issue that we have in deeper term is the cost of screening bottleneck. So why does it hard? The screening bottleneck costs you in four ways. First of all, it's very slow, weeks to months per review, too slow for fast moving areas. And also it's expensive. It consumes a scarce expert time on repetitive data entry. Also, it's inconsistent and you may get inter-reviewer drift and results that are hard to reproduce if you change the reviewers. And it's incomplete sometimes, and relevant studies can easily get missed and conclusion are outdated on arrivals. So this is the bottleneck we set out to remove. And to address those issues, here are solution: AI assisted evidence synthesis with a human in control. And how does it actually works? Uh, we use AI for that. And why we use AI and why now we use AI? There are four reasons for that. First of all, obviously AI reads at a scale, and LLM screens and extract from thousands of abstract in a time. A person can read and can return handful and consistent results. And in terms of consistency, the same criteria apply to identify for every paper. So there is no reviewer drift, there is no bias, and you can get the data as they are. And you can get a structured data from the text. So it turns on a structured abstract into abstract into structured data, so you can compare fields and you can actually analyze them with any tools. And one key reason is recently medical grade AIs evolve very good, and the domain LLM genuinely understand clinical terms, endpoints, dosing, and designs, and can extract the data very accurately. And together with medical LLMs, make exhaustive and structured synthesis practical for the first time. So what medical LLMs unlock in drug research? For example, there are some cases that I explained. First of all, obviously, it brings for you faster decisions. So weeks of evidence review compressed into hours, and you can accelerate it and go calls and repeat the findings again and again to ensure that you get right insights. And also you can do competitive intelligence and try the rivals, trials, mechanisms, doses, and endpoints as the literature evolves. And also it can do earlier safety signals, so surface adverse events across thousands of studies sooner to you. And it is living reviews, so you can rerun same protocol at the moment new evidence or new paper lands and do same analysis on them. So conclusion, never go stale and outdated again. And it is regulatory and HDR ready. So it's structured, cited, auditable packages, and less biased. So you can get more coverage for every paper. And the literature stops being bottleneck and become a living assets using AI and literature review agents. So, what is a literature review agent exactly? It's a guided workspace that runs the entire pipeline for literature analysis and keep the reviewer in control for every step. And it's a platform that AI and experts can work tightly together to conclude reliable results as fast as possible. And it search trusted medical sources, or even you can upload your files in S3 or as a zip, and it can analyze on your sets of paper, or it can connect to external sources like PubMed, Semantic Scholar, any other external sources that you want. And it can screens against your inclusion and exclusion criteria precisely to include or exclude articles from analysis. And also it can extract any data points and any values that you want to get from the papers. Also, it can help you with quickly doing literature QA and answer your question based on the analyzed results. And it is 5 steps workflow. First of all, start with search. So you do query on trusted sources, and you can do, for example, mesh keyword expansion to expand your early queries to cover wider and broader sets of papers. And then you can define the criteria for data points, inclusion, exclusion, and anything. And then you will run the process, and it may take from few minutes to few hours based on your size of samples. And after that, you can select the evidence pack you want, and you can do QA on them and ask any question to reach some conclusion. Also, you can get the raw results as CSV, so you can insert it in any other business intelligence tool or any analysis platform and analyze the data, which will be the second part of this webinar. First of all, let's start with the search. On search, you can search on multiple sources like PubMed, Semantic Scholar, Europe PMC, OpenAlex, and with over 250 million works. And also you can upload your own files. And if you feel that you need to connect it to any other system, it can be connected as long as they offer some APIs to get access. And also there is three modes of searches. You can do old-fashioned keyword searches, which is widely acceptable. Also, you can do semantic searches using vector DBs and get similar articles to a topic. And also you can filter the documents by document ID, or even you can upload your own sets of documents. And you can do query expansion with mesh-aware expansions. And also it offers you a wide range of filters from date, journal, type, language, open accessibility, and citations for filtering low quality papers. And also there is a technique worth naming. Actually, it's a trade-off. So recall versus precision. A broad keyword search maximizes your recall, and you won't miss any single paper in cost of losing the precision for the accuracy of papers that related to your query. So this tool give you option to select between recall and precision by setting the filters. And also in the next step, you define once your criteria from inclusion exclusion to data points, and it will run on the every single papers uh, same like that. And also if you feel that you cannot for example determine inclusion and exclusion very explicitly, here the agent can help you, and you can just write inclusion, exclusion, and data points in natural language, and it will convert it to data points and inclusion exclusion for you. I will show that in the live demo in few minutes. And the extraction and the synthesis engine is the third step, which is actually the core of the product. And what it do under the hood, it uses a medical grade LLM to extract a schema enforced output. So you every time get a consistent schema as output, and it will do automate a screening and do implies inclusion exclusion rules, and also extract the data points that you want. And also it do grounded RAG QA, so you can ask exactly from the literatures and reach some conclusion as some early answer, and later you can verify it against the larger corpus of data. And it's resilient at a scale, so it can connect to any system with any rate limit and any size of papers that you have. And also it's self-hosted, and your data never leaves your infrastructure, and it lives on your own cloud. So it's built for regulated teams by offering self-hosted. As I said, it runs on your own cloud, and queries, documents, results, everything will stay on your infrastructure and will not leave your infrastructure. And it brings JSL or Johnson Labs medical LLMs via Deeplin's API or SageMaker endpoint, so it can deploy it privately for you, so your data never contact third-party LLMs. So you don't be worried about the leaking data. And it's auditable and reproducible, and every result can trace back to a source and rerun on new evidence anytime. And also you can export to CSV, so every steps are transparent. And for regulated teams, these criteria is very important, and this product can satisfy all of those. So let's see an example of it in action. And in this example, we will focus on GLP-1 and incretin therapies in MASH. And we do some early analysis based on that, just an example. Of course, it's not a real project, and it's just some example for demonstrating the power of this tool. So let's start with the case study. Here's the case study. A high-stake drug evolution question recently trending is which incretin therapies improve MASH or metabolic dysfunction associated with steatohepatitis. It's a serious liver disease, and a major focus right now is on it. So for doing that, for analyzing that, we search PubMed broadly and then auto screening against RCT criteria, extracting some structured outcomes, and query the evidence live and ask some question from it. And we do search, define criteria, run, select, and ask all together, and let's work it out together. So let me switch to live project. Uh, okay, is my screen fine? Okay. So here's the literature review agent workspace look like, and it contains five steps. First of all, search articles, define data points and criteria, run and monitor, and two optional sections for select literature and doing literature question answering. At the first page, you can select the source. So you can select from Deep Lens Medical Knowledge Bases, NCBI PubMed, your PMC, OpenAlex, Semantic Scholar, and even you can upload your own files like this. And for example, on Deep Lens, you can select the knowledge base you want. You can select, it should be search MDPI, medRxiv, bioRxiv, PubMed, or Semantic Scholar or not. And you can write your query. Then you can try to expand the query to get broader results. And also you can do different type of search. You can do keyword search or semantic search. And if you select the semantic search, you have ability to MMR re-rank to control the diversity and the relevancy of the documents. So you can adjust between recall and precision. And you can define the date range, maximum result that it should return. You can limit the process for demo runs, and also you can insert the minimum H index that required for the papers. You can sort in different ways. You can select if to search only on publicly available articles or abstract only papers. You can select the author's name, and also you can filter domains, journal names, article type, language, etc. And on PubMed, you have, of course, less filters, but it works at same. So because the PubMed is most updated and most recent medical source, let's just try with that. So first of all, we insert the query that we have. I prepared a query in advance, so it's compares. It's looking for GLP-1 keyword and semaglutide, tirzepatide, and other incretins, and also different forms of MASH, or the older term for MASH was NASH, on also the complete form of the disease name, and etc. So you can use it or expand it. So keep data set small. I just don't expand it and go to next step. And on next step, you can here define what your criteria should be for inclusion, exclusion. And after you select the generate with AI, it will generate the fields for you. For example, here we have for data points, we want to extract drug and dosage. We want to extract drug class, comparator, study design, sample size, population, etc. And for each one, we explicitly say LLM what we are looking for. For example, for dosage, we mentioned that which GLP-1 in or incretin drugs was tested and at what dosage. So it's LLM understand to be looking for those for only GLP-1 or incretin drugs, and any other dosage in the paper will be not considered. Or for drug class, we do same. So we say we are looking for drug class that is a GLP-1 receptor agonist or dual GIP, GLP-1 or GLP-1 glucagon, and etc. And for inclusion exclusion so we ensure that the papers that we want is right selected, we do define some criteria. For example, first of all, we want the papers be RCT or randomized controlled trials in adult humans. And also it should be only for MASH and MASH population, and patient with MASH, older terms, NASH, MA, SLD, and other. And in creating intervention should be mentioned in the paper, and any other papers should not be included. And it should be related to liver and metabolic endpoint because we are interested only on that. And on the exclusion, we don't want non-RCT papers, and protocol only papers also is not useful for us, and studies in animals and in vitros also is not what we are looking. And for the minors, we don't want any papers of that because our main focus is on human adults. So we go to next step, and in here we just need to click run review, and after a few seconds it goes to queue, and in the next steps it goes to listing the articles. And after a few minutes we can see the all articles with the results here as the process goes on. So I have already run analysis that we continue from that because this process will take almost 30 minutes and to avoid delays. So let's continue from that. But the results and the studies are same, except I guess this new article that recently had. Okay. So for here, we have the results for 1,405 different papers, and it's processed all of those, and there was 0 failures. If there was any failures, it will mention here, so you can check that paper manually. And also the completeness is 100. So let's see the results. On the first result, we see the first article is excluded, and there was no endpoint to that extracted. So we check the article. You can see published date, authors, journal, and other fields, and you can see the abstract of the article, and also you can see the data points. So there was no drug and dose in the abstract for the incretin or GLP. And we go to inclusions, and we see this paper was not RCT. That's why it's get filtered out. And if you see on the exclusion, same. It mentioned that the summary is the epidemiological links, and it's not RCT. So there was no protocol in vitro and etc. So let's go to another example. For example, this example is interesting because we have drug and dosage. For example, here is mentioned semaglutide, but there was no explicitly dosage, and the drug class also extracted GLP-1 receptor, and also sample size extracted, and the evidence was in the text here. And for the inclusion, it was RCT, it was a MASH, it was incretin intervention, and liver metabolic. So it's get included and all data also extracted. Another example for the drug and dosage, you can see it here. So you can see it was RCT related to MASH, incretin intervention, liver and metabolic endpoint, and also on exclusion, it was not RCT, it was not protocol only, it's not related to animals, and it was not related to pediatric. So you can see all of these and check all of that together. And let me just change my screen to the CSV result that you can export from the system. So if you click on the download CSV, you will see similar file. It contains all rows and all articles in a structured format, and you have a study title, authors, year, date, journal name, source links for different standard paper IDs, for example, DOI, PubMed, PMC ID, etc. And it contains the summary of each article, full text of articles if available, the data point, extraction result as a JSON format, and inclusion result as JSON format, exclusion result as JSON format, and the final conclusion if this should be included, if this should be excluded, and the status and the extraction time. So you have all of these, and you can insert this exactly into another system to analyze further, which is the second part of this webinar. Okay, let's switch back to the app. Okay, so here was a download result CSV, and we go to next step. On the next step, you can select any sets of the papers that you want. First of all, on here, you can select any articles that you want to ask question about, or you can simply go to next step. Instead of selecting marked articles as a scope, you can set final included test, which means all included articles that had no exclusion criteria met, or you can just select included without considering the exclusion, or excluded articles, or even you can define by endpoints, data points, sorry. For data points, you can say we want to do a question on papers that had a drug and dosage and drug class, or you can select, for example, liver fat reduction also is important. So it will filter those articles, and you can see here the result. And similar for inclusion and exclusion, you can select different exclusion or exclusion should be there or not. Usually we use final including set to ask some question. And in here, you can see the QA environment. So you can ask from the agent, just like what you use from ChatGPT and the similar chatbots. So for here, we just ask which incretin therapies works best for MASH and how do they compare? It's a very generic question. Of course, experts have more precise question for this. And you can see the agent starts to answering. And for each sentences that the agent generates, it brings some evidences. So based on the each sentence, you can click at the end of the sentence on the citation, and see exactly what papers it refers to and use to support this sentence. So every sentence is supported by citation, and you can verify and double check and fact check those to ensure that the results uh, make sense. And also you can check the references, that all top references that use for generating this answer, usually top 15 references you will see here. And of course, other articles are related, but it's answered based on the top articles. So agents review all of those and pick the best and most relatable papers. So you can continue the uh, conversation, for example, with what is teaser peptides, and it will answer you. So you can just continue the conversation and reach some conclusion. Of course, this is for only early conclusion, and for deep analysis, you need to analyze the data itself. So let's go back to a slide and continue from there. Okay. So in the example you saw, we use same questions, and we did research on more than 1,400 articles and around 1,100 papers easily excluded, and 260 articles were considered as final dataset. And as you saw in the single run, we exported data in the CSV, and from the CSV, after analyzing that, you can reach to some conclusion like this. Of course, it's not real examples, so you shouldn't rely on this result. Obviously, it's just case study and example of the system. But as a conclusion, you can see from the abstract, we reach to auditable data set, and semaglutide is essence, and nearly 1,200 patients, and around 62% were using that, and for MASH resolution. So also you see that the second part was other drug, and the third one and the fourth one. So it's just a sample run, and you can conclude the result as any data auditable data set that you want. So here, as a bottom line of what literature review agent do, it's compressed weeks of review in just few minutes and few hours. So it gives you more flexibility to work with data more, and also you get complete structured, cited, and auditable evidence base with a human in control, and your data is never leaving your cloud. If you want to try this product, it's now available on AWS Marketplace. And if you like to try it yourself as demo, please contact support@johnslabs.com, and you will get access to demo instance, so you can try it yourself. And if you had any feedback, please share with us, and you can bring it up on your cloud environment using the cloud marketplaces. That's all for literature review agent. Thank you, and let's go to the second part of the webinar. So far, we focus on turning the medical literature into a structured, cited, evidence-based results. But the value does not stop when review is complete. The next question is how that evidence can support wider drug evaluation progress, and how we analyze them. comparing therapies, reviewing risk, adding clinical context, and placing the result where a decision is being made. So this is where Janus Labs and AI Squared come together. And I will now hand it over to Ian to explain how structured medical data and expert models and governed AI delivery can work as one connected system. Uh, Ian, so it's up to you. Thank you.
Perfect. Thank you so much. So now we have a structured, cited, high signal corpus of medical literature. But how are clinicians actually going to kind of access the the essence of what exactly they need given the questions that they come across in the context of their day-to-day workflow? So thinking about it as more of a human factors engineering problem. So the problem here is that clinicians lack real-time predictive insights into both medications and their suitability for their patients. So there could be frictions, say wasted time referring back to reference literature when there's potentially a short timeline within which to deliver the highest quality care possible. So the solution to this is, of course, you know, getting the output from the literature review system, getting other relevant data points, and bringing those together into kind of a streamlined experience for the clinician, such that it doesn't necessarily replace existing workflows, but that it merely augments them. So I'll take a step back here and let's just think about what kinds of AI ML solutions could potentially provide that support to clinicians. So naturally, not every model excels at every task. And the choice of what model to use can have real implications for the cost as well as the performance of an agentic system. So, for example, the John Snow Labs small medical LLM that we'll show in our portion of the live demo in just a few minutes actually beats out some frontier models on, say, structured information extraction from medical documents. Now, you'd probably want to use that versus, say, like a more expensive, frontier state-of-the-art model like Claude Opus 4.8 when we care about the veracity of information that we're extracting from medical documents, as well as, potentially ensuring that the models that we're interacting with are available within security boundaries that we're placing around the data system that's built to support the clinician's workflows. So the challenge is really how do we bring different AI modalities to bear to solve problems in the clinical workflow. So ideally, you'd have potentially, the John Snow Labs model deployed somewhere to handle kind of domain-specific questions. You'd also potentially be running, complex multi-agent complex multi-agent applications that are making use of AI models across the size and capabilities gamut. So how do we kind of build out a control plane such that we're using the model that we want for the application that we want, given the context that we want? So the solution that AISquared's come up with there is what we call intelligent routing. So given the content of a user's interaction with the AISquared unified platform, we have the ability to actually route the request to specific models probabilistically, as well as deterministically. So say we want to wire a specific LLM into a specific application, we can do so. However, we can also fall back on general kind of flexible logic to make sure that we're hitting like the cost performance Pareto optimal for a given use case. So we've got the right model. Now we just need the right context. So as we saw in the previous demonstration, we now have the ability to extract all of this clean information from, the ocean of medical literature. But a clinician in the context of their day-to-day, they might also care about, information coming from an EMR or an EHR, as well as potentially, say, the pharmacy stock keeping system to make sure that we're able to provide kind of clear and concise information, but also timely, actionable, and accurate information. So this data can come from, many different kind of sources within the hospital's IT infrastructure. So we mentioned EMR, EHR. They're going to be structured relational databases as well as likely relationship databases, so graph databases. And these can just kind of sprawl. And really what the AI system needs at the end of the day is a subset of data, potentially from each, aggregated in context, such that the AI system can actually, make the right, best informed decision. When I say context, that typically gets associated with, LLM context windows. But what we care about here is also the context of the actual physician themselves. So given just an AI output, uh, we see a propensity to either, rarely to blindly accept the output of the AI system, but there can be some skepticism sometimes. So is this Gen AI output actually correct given my patient, given their therapeutic regimes, etc.? So the ability to combine AI outputs with just more traditional like reverse ETL style data in the context of like a chart application can help kind of ground the AI's output and say, okay, this is the information that was presented to the AI system. Therefore, I can sort of understand why the Gen AI system says this, and this. So we've got our models. We've got the information that we're going to pass on to the model as well as to the human in the loop. Now, how do we actually pass that information along? So the fact of the matter is that workflows really kind of evolve over time. I mean, they're something that, somebody's probably using 9 a.m. on Monday to 5 p.m. on Friday, potentially for years. They're a little bit rigid. So take a moment and think, given the awesome demo that we just saw, given potentially the other data sources that we'd care about in a clinical context, how would you kind of package all of that up and give it to a clinical decision maker? So it might be another dashboard, it could be a web app. Now, the challenge is that, clinicians typically have, upwards of 15, 20, I've seen probably 50 open tabs at any given time. Uh, dashboard fa dashboard fatigue is real. And, adding new tools and capabilities that are, optional extras on top of somebody's workflow, that's a good way to ensure that, it's not necessarily going to ultimately be adopted by the end user. So the amazing technology that's been developed, ultimately, ultimately there is a delta between real ROI and, potential ROI. The solution that we've arrived at, and I'll show you in just a moment, is what we call embedded delivery. So taking the synthesized outputs from LLMs and reverse ETL jobs and packaging that up, and again, not supplanting the existing workflows, but rather adding additional kind of views and what we call data apps to kind of go alongside the workflow, sort of like a like a sort of like a dashboard co-pilot, if you will, like an extension that adds information on top of the applications that people are currently using to do the bulk of their job. So we mentioned that there's kind of a sprawl of potential models that you would want to power various different applications. We also mentioned that there are potentially many different sources of information that we'd care about in the context of, supporting, well, really an entire healthcare systems um, kind of span of use cases. So we're talking specifically about clinicians in the context of this presentation, but you'd also potentially have relevant information coming out of or going into like a ServiceNow or an inventory management application. And you would want to make sure that that data is accessible to other systems, but also that you can bring the outputs from AI systems into those applications as well. So kind of a bi-directional flow of data and AI insights. Now, the AI squared unified platform is actually set up for exactly that. So here is just an example wireframe of the solution that I'll demo in just a moment, kind of showing a snapshot of just a single use case where we've got our John Snow Labs small medical LLM that's just deployed in our AWS environment. We've got a Neo4j instance containing data from OpenFDA, and we've got a structured relational database, so just a Postgres instance containing some synthetic patient data. And the Unify platform is connecting that into a mock-up of a clinical workflow app. So just a generic mock-up, and I leave it to you to sort of superimpose, your EMR or EHR of choice on top of that. Now, again, though, this is really more of a control plane for enterprise data and AI. So just like this is one use case, you might have, multiplicative downstream applications. You might have many models, powering many different apps. You might have many different data sources. And the Unify platform is just a way to kind of bridge those gaps, connect the AI with the data, with the applications across the organization. And just like the John Snow Labs solution that we just saw, this can also be packaged up and deployed either on-prem or in a private cloud. So with that, let me go ahead and close the slideshow and open up our live demo. So coming here into our demo environment, and again, this is just a mock-up of, generic clinical workflow application. So again, this is not meant to be a standalone app in and of itself, just to kind of represent, what the power of the right model and the right data can do in the context of a clinical workflow. So we have two tabs here, so drugs and patients. So again, our medication information is coming from a Neo4j instance, and that's just information that was scraped from OpenFDA. And over here in patients, so this data is coming from a Postgres instance, and this is all, just to double click on this is all synthetic patient data. So let's go ahead and click on this first medication. And this is just a blank page, but what you can see here is this inset iframe with the generating output tooltip. So this is this is how AI squared can connect data and AI insights into downstream applications. This is what we call an AI squared data app, which is essentially just embeddable code that connects the front end of your web app with with those databases and AI models that are ultimately going to drive kind of the logic of an application. So here we load it into this medications page. As soon as we did that, this data app started to work. And so it reached out to our medication database, pulled in some of that relevant data, and passed it to our John Snow Labs model that was running on the AI squared cloud. So some of that output from, or I should say, some of that information from the Neo4j instance, we can just use that as is. I mean, that's valuable. So we can come down here, for example, we can see active ingredients, see what conditions it's associated with, what adverse reactions there are. And that's all sitting alongside the AI output from that John Snow Labs model. So what that's giving me is kind of contextualized information about this medication, as well as just a brief kind of TLDR analysis of the medication. Now, what if we care about, say, comparing this drug with an alternative? So, believe aspirin was on the list of similar drugs. So let's go ahead and just compare this drug with aspirin. And again, I'll just hit compare drug. And what we see is another AI squared data app start to run. And so this is similarly reaching out to our Neo4j database, pulling back information about the acetaminophen variant, information about aspirin. It's synthesizing that data via the John Snow Labs model. And what we're getting here is our AI generated analysis, as well as our kind of structured drug data down here. So let's go back out to Explorer, and now let's pick on patients. So we'll choose this first patient here, and again, the exact same thing. So imagine we're in our EMR and we're tabbing between different real patients. The only addition to that workflow is going to be an AI squared data app that can be embedded alongside that data. It could be it can be completely customized in terms of UI UX. All it's there to do is provide kind of contextual, actionable information within that workflow. So here, the data app reached out to our Postgres instance. It's grabbing information about this individual. It's again pulling that back through the Unify platform and passing it to the John Snow Labs model, where the John Snow Labs model is synthesizing an overview of this patient, and then we're taking the some of that structured information about the patient as well as that AI synthesis, and we're pushing that back to the front end. So we'll scroll down a little bit here and we can see this patient's being treated for a common cold. He's suffering from nasal congestion and headache. So let's um let's see what potential medications could help this individual. So I vaguely remember aspirin could help with headaches. So let's see if this could be a potential let's see if this could be a potential replacement for the current acetaminophen regime he's on. And again, our data app here, this is a different data app, is pulling data from our Postgres instance, pulling data from our Neo4j instance, passing that information about aspirin, about acetaminophen, about this patient to that John Snow Labs model, where we're synthesizing a clear, coherent, actionable response. And um, and this John Snow Labs model is just running again, in our private cloud on an A10G, so a very, very low-end previous generation piece of hardware. And we can see after generation, it looks like this is that replacing our current acetaminophen regime with aspirin is a bad replacement. And here we can see a quick synopsis as to why, as well as, additional just vital data about the patient. Now, we can also, rather than just evaluate this drug as a replacement, what if we thought of aspirin as just a potential, you know, additional medication that could help with some of the headaches. So so now we're going to evaluate this as an add-on to the current medication regime. So again, our data app is churning. It's pulling our data from the relevant sources. It's passing that all to the John Snow Labs model, and that's going to generate for us a clear AI overview, some AI generated insights that we're going to display to the physician alongside some structured data. So this should finish running any moment now. And it looks like this is neither a good replacement nor a good add-on. So here's some information that our LLM has output regarding why this is a bad add-on, and some additional information, including the risk factor that adding aspirin introduces a significant risk of um, GI complications, which, given that this patient's currently being treated for a cold, seems like an unacceptable potential side effect. So with that, I'll stop sharing the demo and we'll just jump back to the slides. And so today we moved from a manual evidence review process that can take weeks potentially to an AI-assisted workflow that can search, screen, extract, and synthesize information in far less time while keeping the reviewer in control. We then showed how that structured evidence, as well as other information coming from additional data sources, can support a broader medication insight workflow with the right model, the right context, and of course, the right delivery at the point of work inside of some of those pre-existing clinician workflows. The main point is simple. Faster review only matters when the result is trustworthy, usable, and connected to the next decision. So thank you so much for joining us, and we will now move on to the live Q&A.